Well, DNA. I'm sure we've all heard about it when dealing with forensics, and it's important to know how it's used in forensics if you want to study forensics. The concept of DNA as evidence has changed the whole face of forensic proof when trying to solve crimes or when trying to show someone's innocent or guilty. So it's very important to learn about DNA. We've divided the lectures into two different parts. This part, the first part, is where we just explain what DNA is. And the next part is explained how it's used, how it's collected, how it's used in forensic analysis. So let's get right into it. What is DNA? Well, it stands for deoxyribonucleic acid. There's the D, there's the N, and there's the A. DNA, deoxyribonucleic acid. So we start with deoxyribose. This is ribose. It's what they call a five-membered sugar. Never mind that. It's a common biological molecule. When you remove that oxygen from it, it's deoxyribose, and you have this. Okay, this is the deoxyribose. It's nucleic acid, and because it is acidic, never mind why, but it's acidic and it's found in the nucleus. So that's why we get the deoxyribonucleic acid. It is a polymer, which means it's composed of parts that are pretty much the same, and thousands and millions, in this case billions, of these very similar parts that are made by linking the parts together. That's what makes it a polymer. It contains deoxyribose, a phosphate linkage. This is what sticks the different monomers together, the phosphate linker, or the phosphate linkage, if you want to, whatever. Also, it has, this is very important, the base group. These are bases, and it's ironic that the main part is the bases, and it's called deoxyribonucleic acid. But just bear with me. These are called base groups. They are by themselves basic, and there are only 4 of them in DNA: adenine, thymine, guanine, and cytosine. They're designated by their letters A, T, G, and C. So these are the 4 bases. They're linked by the phosphate, and each of the bases are stuck to deoxyribose. Let's take a look at this, because it's hard to see without actually looking at it. Hard to understand without looking at it. So here we go. This is what happens. This is what it looks like. Here we have the phosphate that links all these things on it. You can consider it like a charm bracelet, where the bracelet part are the phosphates, and the things dangling off of the charm bracelet, the charms, would be the deoxyribose connected to the base. And remember, here's one base: cytosine, guanine, thymine, adenine. These are the 4 bases that are dangling on the phosphate chain. That's what DNA is made of. Again, it's phosphate chain, deoxyribose connected to some base. These bases vary. You can have C, G, T, and A, but you can have C, C, T, G if you want. And there are many, as I said, there are billions of these in one DNA molecule, so they have all different kinds of arrangements of A, T, A, G, C, G, D, well, not D, T. Anyway, so this thing, by the way, this is only one, it's like the half of the standard DNA. There's another strand that is connected on the other side. It forms a helical structure. We're going to see a little bit more about this. Okay, here's really what it, again, a better look of what it is in a biological form. Okay, again we have these phosphate linkers, the deoxyribose that is bonded to the base pair. Here it's C, G, T, and A on this side. But this base pair, the cytosine, will connect to a guanine on a complementary strand. This is like a strand, because it's like a long string of base after base after base. So there is always another string, not always, but in the biological environment, it pretty much always has a complementary string, where if this is a C, exactly opposite of that would be a G. And if this is a T, exactly opposite of that would be an A. A and T stick together. T and A, and here's the adenine sticks to thymine. Here's the thymine sticking to an adenine. They complement each other and they stick to each other very well. They form what they call double-stranded DNA because they're two strands and they're stuck together pretty well. Here we have guanine, C sticks to G, and here's again, C again sticks to G. Whereas G sticks to C, C to G. They all stick together. And the rule is A to G, always, A to G. If you have T on one side, you got to have an A on the other one. And the G sticks to the A. The guanine sticks to the cytosine, always like that. They always have the same complement. Now, and as I mentioned, we have billions of these little bases and with a complementary strand. Billions of them. Now, people say that human beings are the most advanced life form on the planet, the most complex and advanced. Now, you might have heard that DNA is like the blueprint for the organism. So you would think that the most complex organism would have the most longest DNA with the most instructions, most parts of the blueprint. But it's not true. Human beings do not at all have the largest amount of DNA. Frogs have way more than we do. Now, does that mean frogs are more advanced than we are and we should all hail to the frog prince? Well, obviously not, unless you feel like it. Because wheat plants also have more than people. I think they have more than frogs too. And that's getting ridiculous. So let's just continue. It's just something to think about, the length of DNA as opposed relative to the complexity of the organism. Anyway, so DNA, it's found in the nucleus of every cell. I mean, that's DNA, deoxyribonucleic acid. It's found in the nucleus. Okay, that's simple enough. It's found in the nucleus, and it's called the plan or the blueprint of an organism. The way these base pairs are arranged, let's say A, T, G, C, O, this arrangement, that gives instructions to certain proteins to be made or certain signals to be given out. So you have billions of them. You have plenty of room for all different kinds of instructions, and that's what DNA is composed of: instructions, a plan, a blueprint of what the organism should be and what the organism should do. Again, this is on a physical level. It's found in every cell nucleus, and curiously enough, it's found in the mitochondria of every cell, not just the nucleus. This is called mitochondrial DNA. It's slightly different. The mitochondria is the part of the cell that produces its energy. It's not like the brain of the cell, which is the nucleus. It's the part that produces the cell's energy. Why DNA is there is a question unto itself, but it is found there, and that DNA, believe it or not, is actually used, or it can be used. But we're really looking at the DNA that is found in the nucleus. DNA decides, what our features are. Like if we have brown eyes, it's because our DNA says make brown eyes. So we're all different. Therefore, each person's DNA is different. So if we want to examine cellular material to find an individual characteristic, DNA is certainly a useful thing. Now, DNA is critical in reproduction of every form of life that we know. So the way it is, you take a DNA strand. The strand is like a continuous line of all these billions of base pairs. Part of that strand is called that we understand identifies a characteristic. Let's say make a protein, or let's say fast